coccobacillus, which is a little bitty short, fat, egg-shaped instead of hormones. Oxidase negative, except for Plesiomonas. Again, those exceptions. They will all ferment glucose. They all reduce nitrates to nitrite, except two that you'll never see: Photorhabdus and Xenorhabdus. They are all mobile at body temperature, except Klebsiella, Shigella, Yersinia. Most appear on non-selective agar as large, moist, gray columns.
Moist.
Again, exceptions: Klebsiella, Proteus, and some of the Enterobacter. Proteus is the easy exception. We already know what it looks like on non-selective agar. It's going to swarm everywhere. It's also going to stink because those swarmers smell bad. So on like blood agar, we can't really tell. We can tell that it's a gram-negative rock because we know what they look like, but they all pretty much look the same on blood. That's where we have to rely on other tests, because you won't be able to tell an E. coli from a Citrobacter, from a Salmonella, from all of that on blood agar. They're all going to look just like. They're going to be big, moist, gray columns. So this slide, you're going to need to know all of this. General characteristics of the enterics. There are currently, and this is changing daily, 176 different organisms in this group. We're not going to cover all 176. Don't worry about that. So if there's so many exceptions on this page, why were they included in this group? Why would we have the entire group oxidase negative except for one? Because in the past, that was not in this group. The more they do genetic DNA studies and all that on these organisms, they figure out that they're more closely related than their tests lead on, so they start shuffling them around. So they figured out that Plesiomonas, even though it doesn't fit with everything else in the group, genetically it belongs here. So that's why we have these weird exceptions. 20 years ago, Plesiomonas was in its own group. It did not belong in the enterics because of the oxidase reaction. But once they started studying a little bit, they're like, yeah, it's related to those. That's why we have these weird ones thrown in there. And about the time you get these down really, really good, they're going to change them again by next year.
Yeah.
Just like how Group D strips were separated from.
Yep. Same deal.
Yeah.
The Group D, the Enterococcus, they figured out wait a minute, these are different beings, and they started splitting. The more we learn, the stuff gets shuffled around. And they do it frequently. About once a year, they'll move 3 or 4 organisms. Some of them they've moved multiple times. So microscopically, in colonial morphology, again, gram-negative bacilli. Non-spore forming, facultatively anaerobic. Colonial morphology is not helpful. The MAC, XLD, HE, those differential medias, they do help. The selective media contains carbohydrates, which show the ability of the organism to ferment, like MAC. Fermentation indicated by color change of media due to pH change. On MAC, they ferment the lactose, they turn pink. If they're non-fermenters, there's no color change, they're clear colonies. Some of these organisms are your H2S producers. They'll have black centered colonies on HE, SS, and XLD because those media contain the ferric ammonium citrate, that source of iron to produce this black H2S. Classification originally done in 1963. The organisms were all placed in tribes, and then ever since then we've still been shuffling it up. Virulence factors, a lot of it's determined by the organism's ability to stick, produce toxins, invade tissues, etc. Many of them produce antigens that we can use to ID them. The O, H, and K antigens, and then the VI. The VI is only important for one particular organism, Salmonella enterica, with the serotype typhi, which is real nasty. Yeah.
The O's and the H's, they don't have anything to do with the bloodline.
No, totally different. I think they did that on purpose just for confusion's sake. O is a factor of the cell wall, H is the flagella, K is the capsule. And the K, of course, is only going to be found if that organism has a capsule. Not all of them do. Clinical significant, most of these are completely ubiquitous in nature. They're a resident flora of the GI tract of humans and animals, with three big exceptions: Salmonella, Shigella, and Yersinia. That's our pathogens of this group. Remember how I said some things are always pathogen no matter where they're found? This group has three. Most of these are just commensal organisms. They can become opportunistic. Like E. coli, if I do a stool culture on 1 million people, 1 million of them will come up positive for E. coli. It just is. The problem is when you hear like people getting E. coli food poisoning and all that stuff, there's an overgrowth of E. coli. Like if you do a stool culture and all you find is E. coli, that's a problem. You should find 100 different organisms, not one. So speaking of E. coli, originally the tribe was Escherichia. Now that's just the genus. Escherichia coli, and you'll probably never hear that name again. It's E. coli. It's a common flora of the colon. It is used as a primary marker of fecal contamination in water supplies. When they're testing water, they're looking for E. coli to see if any sewage has gotten in it. When they're closing swimming areas at lakes and stuff, they're looking for E. coli. How did all this feces get into the lakes? Animals live there.
People like to dump their
in the summer, as it warms up, it really likes the warm water. So there you go. This is the #1 most common cause of UTIs in humans, primarily in females. And that's simply a proximity problem. Has very distinctive morphology on MAC and EMB. On MAC, usually it's lactose positive. About 90% of E. coli will be pink, surrounded by that pink bile salts. On EMB auger, it usually has a real metallic green sheen to it. Very distinctive to know that green sheen on the EMB. The biochemicals, as listed, endo positive, citrate negative, TSI reaction, all that's there for you. Most of this you can pull from your flow chart as well. Has the O, H, and K antigens. And it goes on to say that O antigen is very similar to the O antigen of Shigella. That's why they're in the same tribe. Yeah.
So these antigens are also virulence factors on here.
Mm-hmm. Very much so. One of the most, and one thing that's not listed, one of the most important virulence factors for E. coli in my experience is speed. E. coli can rapidly divide and grow. So some of these are very slow growing organisms or whatever. E. coli is not. Like in a positive blood culture or whatever, it'll pop up in 6 hours. It multiplies and divides very rapidly. So it tends to become a problem that it can multiply and spread faster than your immune system can fight it. Your book doesn't list that, but I mean, this is one of your faster growing organisms. It's right up there with staph. In an hour, I'll forget what that's at. Again, since most of these are normal flora, they're perfectly fine in their natural site, but we get them anywhere but that natural site, they become a big problem, such as in the urethra, bladder. E. coli wound infections are especially nasty, which, believe it or not, does happen. Especially bottoms of the feet seems to be real common.
For the
Run around barefoot, cut your foot, step in dog poop, whatever. But it happens, and it's a really, really nasty wound infection to get rid of. And there you can see it on McConkey. We should know what this looks like by now. Has the precipitated bile salts around it, nice pretty pink colonies, which means it ferments lactose very well. You can see down at the bottom of this plate, the inactivated E. coli can no longer ferment lactose, clear colonies. Why that's included, I have no idea. But
Why is it inactive?
Uropathogenic E. coli, the strains that cause UTIs usually originate in the large intestine as normal flora. The primary virulence factor is the pili, which allows it to stick to the urinary epithelial cells. The urine flow will not flush these out. Like a lot of UTIs, if you drink enough cranberry juice, eventually you'll flush your system or whatever. It's not going to work with E. coli. Yeah.
Have you heard of E. coli going as high as the ureters?
Yes. All the way to the kidneys. I know a lady that happened to, and the only way they finally got rid of it, silver is toxic to bacteria. They had to insert a small catheter all the way up into her kidneys and inject silver as they pulled out all the way down, and they finally got rid of it.
So,
I'm sure that was painful.
well, she's the one that let the UTI go that far.
Yes. They had to go all the way up.
I didn't know silver was toxic. That's an interesting fact.
Yeah, silver's toxic.
But they treated her with antibiotics for months upon months,
That's my
different
bonus
antibiotics.
case,
Nothing would ever get all of it.
Silver.
They went in with
I'm gonna tell
silver,
him he's
both sides.
toxic.
The gastrointestinal pathogens, there's 5 major categories based on the different virulence factors and the different O and H serotypes. They are all diarrheagenic. Guess what that means?
Okay.
Yeah. Enterotoxigenic E. coli, called ETEC, associated with diarrhea of infants and adults in tropical, subtropical climates, especially in developing countries. Most common cause of diarrheal disease in the U.S. It's called traveler's diarrhea or turista. Anybody know a different name for that?
Montezuma's Revenge?
Montezuma's Revenge. Don't drink the water.
No.
Travelers to developing nations are especially susceptible, usually from consumption of contaminated food or water. Poor hygiene, inadequate sanitation are the factors. Usually self-limiting. Good, normal, healthy people, Montezuma is going to give you diarrhea for 4 or 5 days and you're going to be all right. Usually no vomiting, no fever, it's just strictly watery diarrhea. Symptoms are usually caused by the effects of the toxin released by the organism, not the organism itself. Diagnosis just by the symptoms. And if you isolate solely lactose fermenting organisms on the media, there you go. Again, when you do a stool culture, you're supposed to find E. coli. I think something's wrong if I don't find E. coli. But when you culture it and all you find is E. coli, ding, 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 we have a winner. Because it has taken over. And of course, there are so many different diarrheal illnesses, we have to differentiate from the other ones. So, ETEC, traveler's diarrhea or turista. Enteroinvasive E. coli, EIEC. These are weird ones. They're lactose negative strains of E. coli. They will be clear on McConkey. They cause confusion. Very, very unusual, very rare. They produce dysentery with penetration, invasion, and destruction of the intestinal mucosa. Very similar to Shigella. It's very common to mistake this for Shigella. It is possible to transmit this person to person, fecal oral route, kids, dirty fingers, whatever. Fever, severe abdominal cramps, malaise, watery diarrhea, and diagnosis depends on demonstrating the enteroinvasiveness of the organism. Most labs cannot do this. DNA probe kits are available now, thankfully, because your other way to do it was to go in and rip out a piece of intestinal mucosa and look for it that way.
Oh.
It's a lot easier to do the DNA probe now.
Question.
Yeah.
And the DNA probe, that would be an example of molecular diagnosis.
Molecular diagnostics again, the direction all of the labs are going to. Enteropathogenic, EPEC. This is a long-known cause of infantile diarrhea. Outbreaks used to be fairly common in hospital nurseries and daycare centers. They've improved that a lot now that they know what causes it. They know how to prevent it. Rarely seen in adults. Symptoms: fever, malaise, vomiting, diarrhea. Sounds very similar to everything else, doesn't it? Stool typically contains large amounts of mucus, but there's no apparent blood. There's one difference. Much more mucus is coming out in this. Serological studies usually not done, because diagnosis is based on age and symptoms. If you have a young child with this massive amounts of mucousy diarrhea, we're not going to go any farther than this. We're going to start treating. We're not going to try to ID unless treatment fails. We're going to assume it's EPEC at that point. It says this is what should be suspected in cases of severe diarrhea in children under the age of 1. Just immediately suspect this and start treating. Most of the time you'll be right, and if you're wrong, the treatment will work, you switch to something else. This is the biggie of the group. Enterohemorrhagic E. coli, EHEC. The serotype of this you must know, that's why it's in red. 015787. You'll hear about this a lot in the lab. First recognized in 1982 with an outbreak of hemorrhagic diarrhea and colitis. Disease characterized by low platelet count, hemolytic anemia, kidney failure, watery then bloody diarrhea, abdominal cramps, usually no fever or a very low fever, no white cells in the stool. That's how they differentiate this from Shigella. Potentially fatal, especially in young children and the elderly in nursing homes. A lot of it comes from undercooked hamburger, unpasteurized dairy products, apple cider, bean sprouts, have all been amputated. Recently, kale and romaine lettuce. Do what?
There's always a recall on romaine kale.
Yeah. Produces 2 different toxins, verotoxin 1 and 2. 2 is identical in makeup to a toxin produced by Shigella dysenteriae, Shiga toxin. We can actually, when we suspect this, we can actually do the test looking for Shiga toxin, and this will come up positive as well. And we do that. Other EHEC strains do have Shiga-like toxins and have been found to cause symptoms similar. Diagnosis: use McConkey with sorbitol. That's the only thing that plate is for. Plate has sorbitol instead of lactose. 0157H7 does not ferment sorbitol, and it will appear as colorless colonies. Most of the other E. coli strains will ferment sorbitol. Anything that comes up as clear, you have to test for that 0157H7 antigen using either ELISA or latex agglutination test. This is why almost every micro lab I know of has McConkey with sorbitol plates, and they only use it when they suspect this. They usually expire because they don't use them very often. So usually, if you're doing a stool culture and it is a cup of bloody-looking, watery diarrhea, we'll do the entire plate workup for stool culture, and we'll add a MAC with sorbitol. And the only thing we're looking for with MAC with sorbitol, 0157H7. Know those numbers in that plate, they go together. Yeah.
Okay, so the last part of this slide says also ELISA test now available to detect STA. What is STA?
STX.
Oh, the Shigella toxin.
Yeah,
Yeah.
Shiga toxin.
Thank you.
That's not, yeah, up here, Shiga toxin.
Yeah.
It's not a great abbreviation for Shiga toxin.
They had an episode on Forensic Files about this. to all the kids that were dying.
Yeah, and it has killed a lot of people, and that's why we've started to, and it's policy at National Park, watery stools, automatic macrosorbitol added to it, just from the get-go. If it's solid, you can skip it. But usually if you're doing a stool workup, there's a reason. It's usually watery stools. People have diarrhea and you're trying to figure out why.
Have you ever heard of growing media besides MAC that has sorbitol in it? Or not really?
I'm sure that other plates contain it, but probably not as a sole carbohydrate. If somebody messes up and does not include the sorbitol plate, and you just have the McConkey plate, it's going to look just like regular E. coli. You're going to miss it. And that's why most places have a policy, watery diarrhea, you add that plate. Or some places may have a rapid test for Shiga toxin, and maybe all watery stools get tested for Shiga toxin. But they're going to have something in place.
If the sorbitol media starts turning pink, we know that it's not O1.7.
That's right. We don't know what it is yet, but we know it's not this.
Cool.
And the macrosorbitol looks identical to MAC. It's the same exact looking plate. I think I've actually got some. Enero adherent includes 2 strains associated with 2 kinds of human disease, diarrhea and UTIs. Look at the name, Enero adherent. This is another one that sticks really, really well. You can't flush it out. DAEC, diffusely adherent E. coli, associated both with UTIs, cystitis in children, pyelonephritis in pregnant women, recurrent UTIs. And the lady I'm talking about that got the silver, they think that's what she had. She just constant recurrent UTIs and diarrheal disease. Enero aggravated, causes diarrhea mostly in children. These last 3 we won't care as much about. Those first couple are much more important, especially the hemorrhagic. So the extraintestinal infections. Neonatal septicemia and meningitis. E. coli remains one of the most common causes of septicemia and meningitis among neonates. This is the cause of 40% of cases of gram-negative meningitis. Similar infections are extremely uncommon in older children. Infection usually acquired from vaginal canal at birth. Adult bacteremia, E. coli remains as a clinically significant isolate in blood cultures, may result primarily from GI infection or from GI source. Genitourinary tract infection or GI source. In my experience, someone with a really nasty UTI, which we automatically assume that's probably going to be E. coli, that will jump to the blood faster than anything I've ever seen. The time from bladder to bloodstream is quick. Yeah.
And once it gets in bloodstream, now it's septicemia.
Now it's septicemia and harder to treat. Now it becomes, once it jumps to that bloodstream, which it does very quickly, now you're on a clock. You kill this stuff fast or you're going to kill the patient. Septicemia is hard to come back from. The other species I'm not real concerned about. Escherichia hermani, notorious for being yellow pigmented. Other than that, very, very rare. Vulnerus has been found in wound infections. And Albertii is the newest one associated with diarrhea in children. And from what I can tell, almost everything is associated with diarrhea in children, so just scratch that out of your brain pretty much. Klebsiella, usually found in the GI tract of humans and animals. Absence of motility distinguishes it from most of the other members of the group. Remember, all of them are motile except for 3. This is one of the 3. Klebsiella pneumoniae is the most commonly isolated species. It is distinct because of its capsule. It's a virulence factor that also gives the organism its moist mucoid appearance. This is that one that on McConkey looks like big balls of pink snot, but it's real sticky and mucoid, thick. The reason it's sticky and mucoid and thick is because of that capsule. That's what gives it that appearance. It's a frequent cause of lower respiratory tract infections among hospitalized patients. When I'm doing a sputum or respiratory workup on someone with pneumonia, before I ever set it up, my number one suspect is Klebsiella. Now, it may or may not be Klebsiella, but for whatever reason, this organism likes to go to the lungs. It's a common cause of pneumonia. Especially likes to jump on the immunocompromised, as most organisms do. Newborns, elderly, and seriously ill patients on respirators. Where are a whole lot of your newborns, elderly, and seriously ill on respirators at? Hospitals. That's why we see this so much. Can also cause wound infections. When it does, it's particularly nasty. Can cause UTIs and bacteremia in immunocompromised. There have been nosocomial outbreaks of resistant strains in newborn nurseries. Again, they've learned what caused this, they fixed that. It's not an issue anymore. The other organism, Klebsiella oxytoca, biochemically identical to pneumoniae, except for one test, the endo. And if you look on your flow chart, if you get down to Klebsiella, the next one is endo, and it will differentiate those. Oxytoca is endo positive, pneumoniae is endo negative. So without that endo test, we have no idea which one it is. We know it's Klebsiella at that point, but that's the only way we know for sure. Underlined under pneumoniae, it says usually non-modal. Again, these organisms do not read the book. We say Klebsiella is a non-modal species. 1% or roughly you will find is modal. Extremely rare, just call it non-modal and you're better off.
The only difference between those two is that one.
Yes, they're identical except for that one test. So, and even the machines most of the time won't ID this. They'll ID Klebsiella species, perform endo. You'll do the endo by hand and then you'll put in the ID.
So that's showing us a flag.
Yes. Otherwise, you won't have a species ID. You'll just have a genus. And a lot of people know before they put it in, they see that snotty pink colony, they'll do an endo first, input that data into the machine when they load it, and then the machine already knows, then it'll give you the species. You can do it before or after, but regardless, you've got to do this endo to ID this. Because like I said, even the machine can't tell them apart. And there's your nice, pretty, sticky, snotty looking Klebsiella. You'd think as disgusting and sticky as it looks, that it would probably stink. It really doesn't have a distinctive odor. It's got a smell, but it's not something real distinctive. It's not horrible or anything. It looks like it should be awful.
The plates themselves, they have nothing on the smell.
Huh?
The plates themselves, do they have any kind of scent whatsoever?
Very little. Very little. Yeah.
So this pneumonia, this Klebsiella looks shinier than E. coli.
Yeah. E. coli is usually a very dry colony. This is very wet, moist looking. It looks like the slime kids play with. I know, because I've got it all over my house. It's got little walls in the ceiling. My 4-year-old threw it in the ceiling fan one day and it's like, yeah. We don't do slime in my house anymore. My wife got this big thing. She thought it was fun for the kids to make slime, so every other day she'd bring home contact lens solution, Elmer's glue,
And then every drain in your house is clogged, and so then every drain in your house is clogged.
Yeah.
Yeah.
And then we had to get the glitter glue and the glow in the dark glue.
Yeah.
We had all this multitudes of slime, and it's like, no. I gotta put the dad rule on this one.
It is not fun.
Enterobacter. Colonial colonial, yeah, colonial morphology, it looks very similar to pleb. It's a snotty pink colony as well. Cloacae and Erogenes are the most common isolates. They've been isolated from wounds, urine, blood, CSF. There's a table in your book to distinguish them. Your flow chart will also help. Agglomerans is a complex composed of 13 groups. This one gained nationwide celebrity thanks to being a bunch of contaminated IV fluids. The manufacturer of the IV fluids were not doing their job as they should have, and they distributed all this, and as a result, everyone who got that became septic with Enterobacter.
Oh, no. Like you have one job to do, and you can't fail.
Yeah.
You know.
And they thought they were doing the right thing because these IV fluids, they run them through gamma radiation to make sure they kill everything. Their gamma machine was malfunctioning. Well, of course, there was no outward observation that there was no radiation coming out of this machine. I guess you can stick your head in and see if all your hair falls out, but no one was willing to do that. So they turned all this out, and it actually wasn't irradiated. They thought it was irradiated. Now there's something on the bag that changes colors in the presence of radiation or whatever, so now when they come out, they know. And that changed as a result of this. Here's one they've changed the name of since this PowerPoint was made. Enterobacter sakazakii is now a Chronobacter. And no, you don't need to know that one. This one, they have found it in the powdered formula.
Excuse me.
Until they figured out that they probably need to irradiate that stuff too. And the other two you're probably never going to see, so I don't care about it. Or Hormacii and Asburii. One of my favorites. Only because it's pretty. Anybody want to try saying that name? Pantawea. Pantawea. Pantawea agglomerans. That picture is there only because I think it's pretty.
That's awesome.
Yeah. I thought it was a real shame not to include Pantawea. Yeah.
Is that swarming?
No, it's not swarming. It's just big,
like
beautiful
they-
yellow colonies.
I feel like they did a spiral.
Same thing. That's how they set it up like that.
Oh,
Cool.
Also included, because of something I told you about there, auger art. That's the yellow ink. There's a competition every year where they do art with auger. So you take all these organisms that produce different colors and you like draw a picture. And you can't even see the ink until tomorrow. But Google that, and some of them are gorgeous.
Really?
Yeah, I love looking at it all the time. There's
That's
this
pretty
one-
cool.
So,
There's
this
this woman
yellow
that
ink, they use Pantawea a lot.
There's this woman that
This
does
is your red
it,
ink.
and she looks like holiday.
Serratia.
That's cool.
Opportunistic pathogen associated with nosocomial infections, of which there are 8 species. All of them will ferment lactose, but slowly, except for that one, Ponticola. They are known for their resistance to a wide range of antimicrobials. This is not a common infection organism, but when it is, it's a problem because it's so inherently resistant to everything. The one you're going to see most of the time is Serratia marcescens. Will often produce a characteristic brick red color, even more red in color if you incubate at room temperature. This is your red ink on auger art. Marcescens is a species usually considered clinically important. It's the most frequently found in hospital-acquired infections of urinary or respiratory tract infections, and in bacteremic outbreaks in nurseries, cardiac surgeries, and burn units. Burn units are problematic in a lot of ways. They're almost considered, hospitals that have them, they consider them almost a research lab. That is a complete clean area. Like you're sterilized at the door before and after. They don't allow visitors because burn victims with all this open skin, even stuff in the air, can cause massive problems. They tend to have extensive air circulation filtration units because they're such a problem. The last one, odorifera, notorious only for stinking. Smells like rotten potatoes. Has been isolated from blood, respiratory, and CSF specimens. I've never actually seen it, but that's another one of those with a distinctive aroma. Remember, you always want to know anything distinctive about organisms. That's one of them. So Serratia is notorious for being bright red, and that one is notorious for smelling like rotten potatoes. So there's so much to these organisms. When you can find something that's really distinctive about it, latch onto it. And here's that bright red, brick red color of Serratia.
We did that micro. All the things.
Serratia? Yeah, Serratia's pretty. Again, your agar art, there's your red ink.
We had Serratia, we had B series, and another one.
Marsaceans, for whatever reason, the reason this picture is here, it likes MAC. A lot of times it won't show the red color on chocolate or blood. But you put it on the MAC and it's happy and it kicks out that red pigment. It doesn't, you don't always get that red pigment out of it, which is very disappointing. And again, Marsaceans tends to go red more than the other species. This is another species of Serratia on MAC, and they just turn pink. So even when you get Marsaceans, you don't always get that red. But a really good, healthy, happy strain, you'll get that red color. And you get it more so incubated at room temperature again. So again, distinctive characteristics. Red color goes with Serratia. And most of the time, if you see an exam or textbook or whatever, it'll say brick red. Hafnia, composed of one species but a couple of biotypes. Clinically significant to me because it grows in the beer wort breweries. Has occasionally been isolated from stool cultures, not really known to cause gastroenteritis. Has a characteristics, it's delayed positive citrate reaction. So why is it even included? Because even though we occasionally find it from stool cultures, we're not really sure if that's causing problems or if it's just there. We know that if we treat it, the symptoms go away, but we're not sure if we're killing the Hafnia or if we're killing something else that's actually causing the problems. They suspect this is a GI pathogen. They don't really know because they've also found it in completely healthy people.
Why haven't they isolated it? If they found it in healthy people, could it be considered normal form?
Or heavy drinkers. I mean, I don't know. It's just one of those things that pops up from time to time.
Right.
And they're like, is this causing our problems or not? We're not real sure. And for now, that's still up in the air.
Why did they have to isolate it? I mean, it probably.
I mean, they have. They pulled it out of stool cultures. They pulled it out of stool cultures, people with diarrhea. And they treated it, and the diarrhea went away. But is it because that antibiotic killed Hafnia, or did it kill the E. coli that they didn't find? That was really causing the problem. They don't know. Since it's found in beer, I probably got Hafnia.
I don't like beer.
Big Stinky, Proteus. Opportunistic, normal intestinal flora. The tribe Proteae consists of Proteus, Morganella, Providencia, all of which are PAD positive. PAD positive, Proteus, Providencia, Morganella. Stick that in your head. There are 4 species, 2 of which are widely recognized as human pathogens. Proteus mirabilis, which is the most common, and Proteus vulgaris, which is typically found in urines, wounds, ear, and bacteremic infections. Vulgaris a little bit less commonly than mirabilis, but you'll find them both. Both of these are easily recognized by the characteristic swarming on non-selective media. It goes on to say a brand new species they just recognize also swarms. And the last line I really have a problem with. Swarmer cells produce burnt chocolate odor. That is not burnt chocolate. Textbook, the answer, burnt chocolate goes with Proteus, but I'm going to grow it, you're going to smell it, and you're all going to agree that is not burnt chocolate. The way that it swarms is part of the ascending nature of the Proteus UTIs. They'll start in the urethra, and they'll go from bladder to ureters to kidneys quick, because they can swim upstream. And they swim fast. And that's why this is one, even if you get a UTI with it, it's going to shoot straight up, and it's going all the way to the kidneys.
Question.
Yeah.
It's PAD positive. What does PAD stand for again?
Phenylalanine deaminase.
So on our test, will it be burnt chocolate or whatever it is?
It's going to say burnt chocolate because that's textbook. But again, when you smell it, you're going to agree that it's not burnt chocolate. That is like dead animal in August.
Yeah.
Oh, cool.
Proteus mirabilis versus vulgaris, indole. This is another one of those organisms. Very, very similar in every way, except for this one's just a few tests. It's also different ornithine. But you can see there, they put one spot of Proteus here, and it goes whoop. And I'm going to do the same, probably set it up today. Hopefully we'll look at it in a couple days, and it's probably going to do that, I hope. Because I've got it growing in broth. And I'm going to try to do that same picture. I'm going to put one drop in the middle of chocolate, and we're going to see if it goes. Okay, broth. It's in thio broth. Thioglycolate broth. And again, if you could separate the swarmer cells from the normal cells, apparently the normal non-swarming Proteus doesn't sting. It's the swarming Proteus that does sting. Problem is they're like both the same bacteria, just 2 different types. But apparently the stink comes from the swarmers. I don't know how you would ever separate the different cells of bacteria, but there you go. Figure out where I'm at. All right, we're going to cover this slide and we're going to stop. Morganella only has 1 species. Morganella morganii. There are 2 subspecies. I don't care about you knowing subspecies. It has been documented to cause UTIs. It's also been isolated from other human sites. This is one of those opportunists. Somebody gets immunocompromised, it'll jump on them, but usually you don't see much Morganella. Providencia, kind of the same way. 5 species of this. Brett Gary and Stewartii have been incriminated UTIs and nosocomial outbreaks. Infections of these 2 are especially difficult to treat in immunocompromised because they're resistant to antimicrobials. It also has a species that is found in the kind of Hafnia. It's occasionally found in the stool of children with diarrhea. They're not really sure if that's the cause of agent or if it's just one of those things. When we're talking about resistance to antibiotics, we've already talked about how bacteria can learn to be resistant. Some of these we're talking about now, they're just inherently resistant to this stuff. It's just in their nature to be resistant to a lot of antibiotics. That's the problem with Providencia. It is just naturally resistant to a wide range of antibiotics. Edwardsiella, I don't think it's 3 species anymore. I think it's 5 or 6 now. But Edwardsiella tarda is the only recognized human pathogen. It is an opportunist, causes bacteremia, wound infections. H2S doesn't grow in semen citrate, which is neither here nor there. That's another opportunistic. That's another one that you don't find often. When you do find it, it's problematic. It's hard to get rid of that one. Kind of like the Providencia. You don't see it very often, but when you find it, it usually spells bad things for the patient. Because number one, if they got it in the first place, they're immunocompromised. And now they're immunocompromised and we have a bacteria that's hard to kill. That's just not a great combination for the patient. Usually spells bad things for them. I mean, they've really got to be on the uphill improving to have a chance when they pick up one of these. Yeah, there.
Is Edwardsiella a proteus?
A what?
Is it a proteus?
No. It's in the same tribe. But no, it's not a Proteus. Proteus is a different genus.
So,
Edwardsiella being the genus of this one. Yeah.
so since it's not a proteus, we can assume that it will be PAD negative.
Correct. There are only those three you need to connect with that PAD. Proteus, Providencia, Morganella.
Vulgaris.
Huh?
Vulgaris?
Vulgaris and mirabilis are species of Proteus. Proteus vulgaris, Proteus mirabilis. That is about halfway through this chapter. And that's all I want to dump on your brains at one time, because this is a long chapter with a lot of organisms. It's a hard chapter.